Thank you, Matteo, for the nice introduction. And so let me first share my screen. Okay. Um, I hope everyone can see the screen fine. If not, just let me know. So, thanks everyone for being here, for taking the time to spend an afternoon with us talking about science and art. As Matteo mentioned, I am actually still a scientist. I'm a full-time postdoc, which is what perhaps makes my talk slightly different from what we've had before. Um, but what I really wanted to share with you today was a little bit of my creative process. And the reason why I wanted to do that is that I really think that art and science are not really that different in when it comes down to being creative and making something new that doesn't exist. So first, I would like to start from where do I get my inspiration? Because my inspiration actually comes from my work. So I think being a scientist exposes you to wonderful things, not every day, but really, really often. And this is a collection of images that I've taken during the last 10 years that I spent in different labs during my PhD and during my postdoc. And honestly, like this is probably what drives me in my daily work as a scientist, but also as an illustrator, like seeing beautiful things and seeing how life works at the microscopic level. Nonetheless, I am a molecular biologist by training, and I very much love molecular biology. And the reality of the molecular biology lab is quite different from this slide. It actually looks a little bit more like this. So as you can tell, I'm not the one with the tidy bench, everything clean and perfect. But as a molecular biologist, what I spend most of my days with is tubes, clear liquid, teeny tiny amounts of clear liquids being moved from one tube to another. Sometimes you look down the microscope and you see something exciting. But most of the time, you don't really see what's going on in your tube. And that's where I think imagination plays a really cool part because you have to be able to visualize what's going on in your experiment, in your theory, and in order to actually make things work, you have to really use your creativity to be able to visualize what's happening. So this, I would say, is one of the key drives for me to venture into science illustration. Because when you tell people that are not molecular biologists and they're not scientists, what you do in the lab, what they see is usually the clear tube and maybe, you moving teeny tiny amounts of clear liquids from one tube to another. And, it doesn't mean much to them. But it doesn't necessarily mean that they can't get excited about what you do, because once explain it to them, once you make it visual, which is the key word here, they can actually start seeing what gets you excited and what is actually happening inside of the test tube. So I think I'm a very visual person, like many people here. I've been deeply in love with drawing since I was a child. And so for me, really, what I could say is like sort of my mission is to really like bring to life what is inside of the test tube that I can kind of visualize in my mind in order to convey the excitement that I feel for my work and for what I do to other people, including not scientists, because I think it's really, really important to be able to communicate science effectively, not just to your peers, but also to people outside of the lab. So there's many ways you can do that. We've seen comics just now, which are absolutely amazing. I'm also like really a big fan of comic books. The style, I would say, that I've adopted in my scientific illustration is the use of metaphors. Because I think metaphors, both verbal metaphors and visual metaphors, can be really, really powerful. So if let's say I'm trying to describe to my parents what I'm working on and they are not scientists. If I tell them about, oh, this molecule is doing this, is then doing that, and then we have protein, we have RNA, they're going to be like, what the hell? What are you telling us? Like, what is this? Is all this jargon? It's difficult to grasp if you don't really have, a scientific education. However, a metaphor, if I translate my work into something visual, into something metaphorical that they can grasp, all of a sudden it clicks and they can follow me, they can get excited about what I'm telling them, and they can catch actually the big concept, the overarching concept of that scientific topic I'm describing. So when I go about finding the correct metaphor for a new scientific illustration, I would say there's three key points that I have to fulfill. First of all, it's got to be scientifically accurate. I'm a scientist, I work for scientists, so we don't want bullshit. We wanted to describe what we think is an accurate topic. So we cannot compromise. This is something that we cannot compromise on. Secondly, it's got to be simple. And this is really, really important because most scientists, they really get captured by the small details. They have to be really particular about it having everything. And that's something, it ends up overloading the message. So you really have to remove all the unnecessary bits from your message and strip it down to the bottom to be simple, but also captivating, because you want to be exciting, you want to capture your audience, and you want to engage them. And finally, once you have an accurate metaphor that is also captivating, you can have fun and adding back the details. I always like to put some nerdy details in my illustrations that are maybe, an actual scientific plot that becomes part of the illustration, or maybe some particular shape of a protein or something that you would see down the microscope translated into an element of the illustration. And that's really nice because that's where specialists kind of get excited because they recognize that something and they're like, oh yeah, I know, that's the nucleus of that particular cell, or oh, that's a plot of something, and that's quite fun also for the nerdy scientist that we are. But however, science illustration starts exactly where a scientific project starts, which is from the papers. So that's, I would say, the first similarity that I find between my illustration work and my daily job as a researcher, because you've got to read the papers, you've got to understand them, even though you don't grasp the tiniest details of it, because sometimes I get to illustrate stuff that is quite far from my comfort zone. It is still really important to get a general understanding of the topic you're trying to illustrate. And then the second step is actually the most daunting, but also the most exciting part of the process, and it's the white canvas, because you've got a completely blank space. Most of the time, you're given free reins by the clients. Some people come to you with already a little bit of an idea. Some people are like, you have free reins, do whatever you like. And that's, I think, probably the part I like the most, but also I fear the most, because you've got to come up with an interesting idea, and that's not obvious. And that opens up a range of possibilities and a really wide canvas of the creative process. And when we talk about creativity, that's where I really think that science and science illustration kind of connect. Because in both cases, you can't force yourself to be creative. There's no recipe. I cannot tell you, you do this and that, and you're going to have an amazing project idea for your postdoc, or you're going to have an amazing illustration. But what I would like to share with you is what works for me and how I do it, with no idea of trying to teach you anything, just sharing something that for me is deeply exciting. So what works for me is, for instance, trying to grasp general concept of what I want to draw, and then sort of let it simmer at the back of my mind. Kind of not looking at it directly, but kind of looking at it from a distance and, with the corner of your eye, and let it simmer in those moments where you can have a little bit of free space, because having free mental space is absolutely essential to come up with a good experiment and with a good illustration. Because when you're overloaded by meetings, by experiments to do, by everything that goes on in our daily lives, you can't just come up with the best idea. So you've got to carve some time for yourself to be creative and to let your thoughts somehow run free. And so for me, sometimes, when I have these moments, it can be in the car, or, when taking a walk outside or going for a run, when the brain clears from the daily tasks, that's where I can start thinking about what I want to draw. And once I get the idea that, maybe draws inspirations from things I like, from things I've seen, from nature, from comic books, from, just things that I find aesthetically appealing, then it becomes sort of an obsession, because then I really, really want to turn it into reality and into an art piece. And so I like to define my illustrations as unconventional science illustrations, because I really try to find an unconventional imagery to depict scientific concepts. And so in the next few minutes, I would like to show you some of my I can't say my favorites. It's like, with your children, you're not supposed to have favorites. But, some illustrations that I really, really care about and I really enjoyed doing. So this is one of the first ones I made, and is about a molecular biology process called splicing and the involvement of M6A, this RNA modification. So when M6A is present, and I wanted to depict it as a lamp, you do not have splicing. So you don't have this cut and paste of the RNA that is important to make a mature messenger RNA. However, when the RNA is not methylated, this lamp is fully lit, and it attracts a lot of splice factors that I wanted to draw as moths that are drawn to the light. Another one that was an absolute obsession for me was this one. I really wanted to depict this as stained glass, which is something that I find visually very appealing and very inspiring. And so this was a work from a lab in Cambridge that had developed a bioinformatics tool to interpret the chromosomal instability signatures of cancer that you can see here depicted. These are real plots from ovarian cancer depicted on the windows. And they were able to decode the signatures of this chromosomal instability into pathways. So for instance, when you have this tool, you can shine light through the plots and you start seeing images. You can start seeing DNA damage, which is one of the processes that go wrong in this context. However, when there is no light, so when you don't use this tool, you can see only shadows and, kind of fuzzy images. This other one is also very dear to me because I work on oogenesis, so fertilization is a topic that is very close to my heart. And this group in Vienna had found a protein complex that was able to unlock the door of the oocyte and open up the path to fertilization. And this other one was instead done for a very exciting work that actually I was super happy because this was a paper that made the news worldwide. So I could tell, my friends and my family, look, I've made an illustration for something that actually made the news. And it was in vitro models of human embryonic development. So we incorporated the actual embryo model in the illustration. So in the last few minutes, I just want to answer the question that maybe Matteo nicely introduced me with. So how do you juggle two careers? I do not have an answer. Actually, if you do, please let me know. But I do want to share a few notes from my experience. So there is no way that you can do both at 100% capacity. If you're trying, forget it. The key here for me is in the balance. You will know, your gut feeling will tell you what the right balance is at every point in your career. There will be a time when you have more open space, open time to devote to your illustrations. There will be times when you're really swamped with deadlines and you have to prioritize science. But as long as you enjoy doing both, which is the case for me, you find the balance. You find a way to do both and to be happy and fulfilled with both. There might come a time when one has to choose, but really, if you want to do both, you can just tweak your balance depending on the time of your career. And with that, I'd like to thank everyone. Thanks, Philip, for organizing, and thanks everybody for being here listening to us this afternoon. And I will pass it on to Radhika Patnala. So she's the founder of Illustrate and Endosymbiont. She's a neuroscientist by training, and she will share with us some of her favorite projects from recent years. Over to you, Radhika.